Welcome to the University of Colorado School of Medicine's Ask an Expert with Dr. Rosemary Rochford.
I'm a professor in the immunology and microbiology department here on campus, and I also have a adjunct appointment over in environmental occupational health in the School of Public Health. And I do research primarily on Epstein-Barr virus and malaria, and work overseas in Kenya and Uganda. So I have more of a field-based research and also have a lab-based research program. So a little bit of a virologist, a little immunology, and a little epidemiology.
Can chloroquine treat COVID-19?
So there's a lot of work in the press about this, but this is always good to go back to the published papers to see what's in the literature, right? And so, I think I told you I work on malaria, and actually, one of the studies I work on is looking at drugs for malaria. And the 8-aminoquinolines. Chloroquine is a 4-aminoquinoline. So, just a little history of the background. And chloroquine was first discovered. If we go back even further, the quinine of the gin and tonic that we love that it was taken as an anti-malarial. Chloroquine. So maybe if everybody drinks gin and tonic, we can just solve the problem, right? Um,
Yeah.
but I'm being facetious about that. So the chloroquine was discovered in the 1930s as a treatment for malaria. And it's a very successful treatment for malaria. It works against the blood stage. It works great, not a problem. However, drug resistance developed to chloroquine and really became a significant problem. And so the efforts to develop other antimalarial drugs came to place, and artemisinin is there now, and so there's other drugs. Um, because of that, there's actually been that loss of that resistance to chloroquine. So it's still used for antimalarial treatments around the world. So it's not that it's not effective. It's just where there's resistance, it's not effective. Um, so the question is, people have been saying, well, if it works for malaria, what does it do for viruses? So the question, can you use it to treat viruses, is not a new question. And even back in the 1960s, they were trying to throw it on virally infected cells in culture. And they did not, they saw some kind of modest effect here and there. And then in the 2000s, after the SARS epidemic, people showed that the SARS-CoV coronavirus, not the one that's CoV-2 that we're dealing with now, that it was inhibitory to replication. So looking at in cell culture and in vitro system. But one thing to think about drug studies is what you do in vitro are very different things, because you have to think about metabolism and the pharmacokinetics. So the dose you can give on cells in culture might be really high to see an effect, but what you can actually get into people is not that same dose level. So you always have to be sure you translate what goes on in vitro to in vivo. So there is data. There's a paper that was just published that shows some effect again in vitro, but they used varroa kidney cells, which are not the same as lung epithelial cells. So again, thinking about what's in vitro, cell culture, long-term cell lines, different biology. When we think about our lung cells, right, they're have an apical and basal side, right? So they have side to oxygen, side to the blood system. In cell culture, very different kind of modeling. You don't get that true biology of those cells in the cell culture system. So we have to be careful what to translate. So it seemed like, I think it's not a bad idea for people to have tested it, but there's only 2 studies that have been done and are out there. And the first one is a Chinese group that published the data on this, but they compounded data from a lot of different studies, and they haven't published the data so anybody can look and see what the primary outcomes are. So we don't have any. It's sort of like, look what we found, except they didn't show you what they found yet. So maybe it's coming. So we really need to see that data first and to understand what the study design was and the appropriate to study design. And then the French study, again, purported to have effects on the viral load, but they didn't look at any other outcomes other than the viral titer. And it was what was called it wasn't a randomized clinical trial. They selected the patients, and it was 24 patients. So when we design new drugs for development and study, we do randomized clinical trials where you take patients that are sick and you randomize them to the treatment arm and the non-treatment arm. And you then look at the data so you're not biasing yourself to sort of take the healthier patients or any kind of biases that we might tend to do, because we're humans. So we try to make sure that we do a trial in a way that is completely unbiased. So because that data was randomized, non-randomized, I mean, so it wasn't, and it was a small group. Again, we have to think about power. You think about humans and our incredible variability. So you need to power a study and have enough people in that study to really understand what the effects are. So Both studies have limitations. The Chinese study, again, we don't have the data to really evaluate it appropriately yet. And the French study, we don't have an adequate sample size yet and an appropriate study design. It wasn't, as I said, it wasn't a randomized clinical trial with just 24 people. So intriguing, but not convincing yet. So um, to take that data and then say I would recommend treatment for a patient seems a little bit premature. Um, and again, it's not that there might not be reasons why it wouldn't work, but again, the data is based on in vitro studies that the dosing was very high in different types of viruses and cells and so on. And then the in vivo human studies, um, we don't have enough data yet to make a conclusion. So if it was me, and I'm not a physician, I'm a PhD, but I wouldn't want to take it. And the other thing is you have to realize is that this drug is still used to treat lupus patients, the hydroxychloroquine version of it, and it's also still available and used around the world for malaria. So the lack of availability because people are rushing out to use it is affecting other people's ability to have access where it's proven to be important for clinical care.
What about azithromycin?
I don't know as much about that, so I can't. I mean, it's an antibiotic, right? So it typically targets bacteria. People know in clinical take a Z-Pak.
Mhm.
Um, so whether that's going to be effective, why a bacterial antibiotic would be effective, I'm not sure. Right? When we think about influenza, the cause of a lot of morbidity and mortality with flu is secondary bacterial infections, right? And that's typically what you take a azithromycin for, right? So you get flu and you get the influenza virus infection, and then you get the secondary bacterial. I'm assuming that might be why they propose to include that. There is some data I saw somewhere else, and again, I don't know as much about azithromycin, but um, it might be because of that, or maybe they had an idea it might have some antiviral effect. I'm just not sure. I might just point out, though, it's an erythromycin derivative, and for those people that are allergic, like myself, there are people that are allergic to that, and so you also have to be careful in thinking about that. It's not one people typically think about penicillin allergies, but certainly it's one that you can develop allergies to.
What are your thoughts on our response to COVID 19?
What I would say is that I think that there are a lot of really smart people all around the world working hard to kind of come up with ways to deal with all aspects of this. So I think that the encouraging thing, and I read this somewhere, is that scientists, physicians and scientists, physician scientists, think globally, right? We always work with our colleagues around the world. I work in Africa. I have friends in Europe. I work over there in South America. So um, this is a problem for the entire world, and it's a problem that we're all thinking about collectively. Um, and I think if we understand that, I hope that we'll come up with some solutions either through therapeutics, antivirals, things to mitigate the disease in the patients, or ultimately a vaccine. But I also think that's going to be a ways down the road because that just there's a lot of work. If we think of go back to understanding of HIV, just to want to get a vaccine doesn't make you get a vaccine. There's a lot of tricks to the trade, and you could ask everybody. And for the med students, I lecture the respiratory virus lectures every year. RSV, we don't have a vaccine for respiratory syncytial virus. It's a big problem. We only have, we provide Respigam for the infants. So is it going to be like respiratory syncytial virus, or is it going to be like influenza virus? And we don't know the answer to that. So I think until we understand more about how the body fights off that infection, we have to we're going to have to hope it's more like the flu than the RSV, but we don't know. There's so many unknowns with this, right? I mean, this virus, and if you think about this virus was just sequenced like in early January, right? So we're 3 months after we've actually discovered a brand new virus, sequenced it, got this testing up and running. So in some ways, I think everybody hopes we're going to be a lot further along, but we've actually gone an incredible amount already with tons of clinical trials around the world. And I think my only thought is to always stick with the science and stick with the data and look at the facts in front of us and not get ahead of ourselves because we want to solve a problem. We got to solve it the good old hard fashioned ways, just get in the lab and get in the clinic and do the right test and have the right controls, right?
How are you keeping busy during self isolation?
Um, I'm working actually. So we're the university has put together through the Department of Medicine, Dr. David Schwartz. We have a consortium working on COVID-19, trying to sort of think about how we pull the faculty and staff and students together to address this within our own community, and if we can also contribute to the larger effort around the world. So that's kept me busy. So I haven't walked away from COVID-19. Although now that the weather's nice, I'm happy to be out in my garden, which will be really that's a nice break from everything.